Welcome you all to this cryo-EM course. Last class we already discussed about how we can glow discharge our grid, and that is for negative staining as well as cryo-EM grid. For both the cases we have to follow the same protocols. So we have to do the glow discharge of our grids. It may be the negative stain and or for cryo-EM, whatever grid we will use it, we have to glow discharge, but glow discharge timing and strength will be different. Generally for negative staining TEM imaging, we glow discharge our grid for 30 seconds, sometime 40 seconds. For cryo-EM grid, it is mainly quantifoil or C-flat grids. I already mentioned that quantifoil grid generally we go for 1 minute glow discharge or 75-second glow discharge based on our protein sample, quality of the glow discharge unit, and what kind of buffer we are using. We may have to change the glow discharge parameters. Glow discharge is an important part when we are doing cryo-electron microscopy or negative staining TEM. If our grids are not hydrophilic, most of the time biological sample will be not well spreaded on top of the grid and our imaging quality will be bad. Not only that, I also discussed how to do the negative staining, and that will be clear when you will see the practical session how to make negative staining grid. Today I am going to discuss more about the good and bad practices for negative staining grid preparation, as well as I will show you today's main part. It will be mainly showing different kind of TEM images. Specifically it will be negative staining room temperature TEM images, and from there what we can interpret. Is it a good image or it is a bad image? What is a good image? What is a bad image? What is the bad image? How we can collect good quality images, that we are going to discuss. And then I will also show you some kind of applications, how we can use TEM to collect a good data, and how we can analyze those data, and from there what kind of conclusion we can make from those TEM images, that I am also going to discuss. Let us first discuss about the negative staining TEM images of soluble protein. Why I am telling soluble protein? Because soluble protein, whatever protein we are going to deal here, that will be a soluble protein. It will stay in the buffer solution. Some cases there are some protein, those proteins are not soluble protein, they will go to the pellet. Mostly some protein expressed in the membrane, and they need the membrane component, and most of the time those membrane protein will go to the pellet, and we have to solubilize those membrane using the detergent. That we are going to discuss in the later stage, how we can solubilize the membrane protein. And membrane protein is one of the very interesting research topic nowadays. So if you just consider any kind of receptor, transporter, pump, all are mostly membrane protein. So all these membrane protein are surrounded by the lipid bilayer. So if we want to isolate those membrane protein, definitely we need a different kind of components to solubilize those membrane protein. If proteins is precipitated, or if it is staying in the pellet, and if it is not solubilized, so it will be not in the solution. If it is not in the solution, it will be very difficult to do the TEM imaging or use those samples to for cryofreezing and do single particle analysis. I already discussed about that. What is single particle analysis? Single particle analysis is your protein will be separated from each other, and there will be multiple protein will be, you will just select individual protein molecules, and then we will analyze those protein molecules, we will classify them into different 2D classes or 3D classes, and from there we will calculate the 3D structure. That is called the single particle analysis. So for the single particle analysis, a protein will go to the pellet or associated with pellet and precipitated within the pellet, it will be very difficult to identify single protein molecules. So definitely it is not going to work. So that is the reason for the membrane protein we have to apply a different technique. We have to solubilize those membrane and isolate the protein. When you will isolate the protein from the membrane, then you will follow the same protocol. So we will discuss about that membrane protein solubilization and how to extract the membrane protein from the membrane and how we can go for the study. But before that, we are going to discuss about the normal room temperature TEM, negative staining of the soluble protein molecules. So let's start with those imaging system, and then we discuss about how we can go, how we can analyze it is a good TEM image or it is a bad TEM image. So now we will pay attention to our slide, and from there we will start our discussion. So if you look at that slide for TEM images, where you can see, so TEM images most of the time whenever we are talking about magnification, we mention it as a scale. The scale bar is very important. Whenever we will collect a TEM images, we always consider that what magnification we are collecting our data. And the magnification, when I discuss about magnification and lens system, I mention these things. Sometimes those things will be little bit variable. That is the reason most of the time we are using a scale bar that is calibrated based on our microscope, camera position. So this calibrated magnification that appears as a scale bar in your micrograph. So whenever you will show any TEM images or cryo-EM images, don't forget, always keep the scale bar. So it may be a nanometer range or micrometer range. Based on your magnification, this will vary. Now here you can see there is one TEM images. Most of the time you can see, so we will discuss about the pixel. So we can split this image at different pixels. So it will be a either 2000 by 2000 or 1K by 1K or 4K by 4K. So generally you will get an image like this. Now if you look at these images, this is basically a negative 10 image. So based on the title that we are discussing about the good and bad negative staining images, right. So here if we just collect an image like this one, and what do you think, is it an good image or bad image? So when we are talking about a good negative staining image, if it is a protein sample, it should be monodispersed and it will be isolated, and you can see the morphology of the protein. Later on we will discuss about the different type of cell imaging as well as the bacterial cell or mammalian cell imaging also. So now if you look at this particular image, what you can see, there is a lot of white patches in different areas, as well as there are some black patches somewhere here in this particular area, in this particular area also. But you can see that it is not very clear from the image what I am trying to see. So initially I will discuss about some bad images. If I just consider this is a good image or bad image, first I want to mention this is definitely a bad image. Okay. So don't consider it is a good image. Why I am considering this is a bad image? Because so if we just look at, there are many places there are some white circular part is there, so something like that, something like this one. So there are different shape of this molecule, but same time if you pay attention, there is some small molecules are there where central part it has some hole. So I am not very sure if it is a new sample. It is a very difficult task from this negative stain image to pinpoint is it a good is it a good image or is or bad image. But definitely from this one I cannot isolate anything, because you can see some places there is a lot of bad lot of large patches are there, and I am not very sure what those patches are. So definitely if you just consider this kind of images, it is a bad image. So whenever we will talk about that, okay, we want to do a TEM imaging, we are using negative staining solution. Negative staining solution means it will be uranyl acetate or uranyl formate or ammonium molybdenate and other staining solution also. Sometime we are using different type of heavy metal staining solution also. So those staining solution, we are always expecting that it should be well distributed and the protein will be well distributed on that. And then if you just think about in this when you are using heavy metal staining solution, the scattering will be much higher from the background and your protein molecule will be less density will be less, and then electron beam can easily go through the protein sample, and then we will get white protein molecules in comparatively black background. Last class I showed one negative stain image, and that time I discussed that where the uranyl acetate or uranyl formate is deposited and how this background is looking. And in this particular image definitely we cannot see any details about the protein molecules. This may be a protein, this may be a protein, this may be a protein, this may be a protein also. So I am not very sure. In that reason we are considering this kind of images are really bad images that we really don't want to use it for any purposes. So if you have this kind of image, we should redo the negative staining. Let us go to the next one. Okay, so now you can see there is a another type of sample where you can see there is a filamentous you can see there is a filamentous protein something that filamentous protein is going. So this is definitely another protein sample and those are going like that. So here we are expecting some kind of trimeric structure. We are not expecting any filamentous structure like this or filamentous structure like this one. So what we expect here, we are expecting something like this one. Now from this negative stain, even after ACC purification, if you recall the ACC purification, what I told you that okay, if this is a ACC profile, if this is your volume, if this is your absorbance, so it will go like that and then it will give you a peak like that. And from standard curve means that you already know the molecular weight of the protein and then you are running from the ACC, so you can generate a standard curve from there. Okay, so at volume 8 ml, 10 ml or 15 ml you will get so you have known protein, and that known protein it is going, so it is eluting here, it is eluting here. From there you can get a standard curve, and if you just look at this standard curve and if your volume is somewhere 9 ml, from there from this standard curve you can calculate what is the molecular weight of your protein. Now you just consider that okay, from this one I determine that my protein molecular weight will be a trimeric and it should be a 200 kilodalton protein. Okay, so here we are expecting some kind of trimeric shape, but in all over that you cannot see any trimeric shape, but your protein is eluted at 9 ml. So we are expecting a proper folding of the protein and give us a trimeric shape protein, but here in this particular image we cannot see that. Then definitely it is not a good sample we can consider. So this is one of the beauty of negative stain, that when you have a purified sample, you take this purified sample and do the negative stain, and from this negative stain you can pinpoint it that okay, is your sample distribution is proper, or sample proper folding, quality, everything is correct or not. From there you can see that. If I ask you, definitely this protein sample, if you just consider stain wise, this image is looking much better than previous one. Previous one, you know, if this particular image here, as I mentioned, there is a lot of heterogeneity. There are some protein like things are there, but rest of the part there is a lot of background. This background means probably stain is not proper. Here staining is comparatively much better, that is why you can see all this helical pattern of that protein, but it is not properly folded. So from this experiment or negative staining experiment, we can tell that protein has the proper morphology or not. And now if you just look at this image once, that you can see there is a black patch here, there is another black patch is here, some another black patches is coming here, and also some black patches are coming this area. Now if you look at those areas, you can see there are multiple these helical things are coming together and then forming a cluster. Here also you can see and cluster, here you can also see and cluster. So probably this protein, it is unfolded and there is a lot of unfolded regions are there, they are coming together and forming some kind of aggregation. So definitely if you just consider negative staining, staining, it is much better than previous one. Previous sample, but overall sample quality, if I just consider that sample quality is bad. So that I told why the sample quality is bad, because I know from my previous experience, because this sample I already imaged previously, from there I am expecting this kind of trimeric or this kind of shape, but I cannot see it here, and that is the reason I am considering this sample is not a good sample for any other study. So definitely we have to go back this particular protein purification stage and we have to perform that protein purification. What about the next sample? Now I am coming to the next sample. If you look at this sample, you can see there is a white some area there is a white background. Here you can see there is a white background, but the background is very prominent here. Definitely whatever stain we did it, it is not very good stain here, but in this particular image you can see there are some isolated particle. Here you have an isolated particle, here we have a isolated particle. So those, there are many places we have isolated particle, and if you look at the shape of this particle, whatever I was showing last image, you can see similar kind of triangular shape molecule here. So if you look at this particular protein, in all the cases you can see there is a this two and then this kind of. So there are many isolated particle where you can see different triangular kind of shape. So that means definitely this is a good sample, and comparatively this image is better than any other images that whatever I showed you in last two images. In one image it is completely very bad stain and you cannot distinguish from that stain that background and your protein sample. There probably stain is not well distributed, that is the reason we cannot see our protein molecules properly. But in this particular case we have a strong background. So, this is, I can say, background, this is the background area. Okay, this is a background area, but definitely if you look at, you can easily isolate many protein molecules from here. That is basically we can consider that this is a much better image than last two sample. So, if you look at any image, first we have to figure it out how my protein should look like. If there is any negative staining term or if there is any published crystal structure or cryo-EM data is there, first you compare is there any images are available or not, and then you compare your images with the other images, and that will give you a better idea is your sample is looking good or bad. If your sample is looking good, then you can compare that, okay, do I my images are looking good or bad. So, in the next image if I just go, you can see it that similar kind of image, and where also I can circle couple of protein molecules here that will give us some kind of idea that we are expecting this kind of triangular shape molecules. So, what I did from here, I just pull out couple of those protein samples and I represented it here. Now, if you look at this, there are many this kind of, wherever you have a black background, these protein molecules are very much isolated and it is looking very prominent. So, what is happening, if you recall your last class what I told you, there is a three type of staining is possible, right. What you have here, you have the carbon and top you have carbon. Now, when you have any protein sample, if you just consider that, okay, this is my protein sample, I will just use that yellow color because yellow color will be not very visible. But now, if I have a staining solution, so something like that, so if I put that staining solution, you can see the protein sample very well, right. So, because it is giving you a proper background. So, giving you a proper background. Now, based on this possibility that there are three possibilities is there, that okay, if we have a carbon coated grid and if I have a sample here, sometime you get a staining solution or stain will deposited like that, and then your protein molecule and then scattering from this stain will be, the staining solution will be that not grid. So, the scattering between this protein and from the background it will be almost same, and then you cannot distinguish your protein sample from the background, and that is basically called light staining sample. So, whenever you are doing negative stain, there is a one term it will come that, okay, thick stain, light stain, moderate stain. So, when we have a light stain means your staining solution is deposited, it is not that thick stain and your protein is somewhere here and the scattering is almost same, and that is one of the reason it is very difficult to distinguish the protein sample from the background. Now, here this is basically, if I just call this is a thin stain, right. I told that, okay, this is a thin stain sample. So, here you just consider that, okay, so in some places wherever the protein sample is visible, if you look at in surroundings area, that is a very black region is there in the background, and that is the reason you can distinguish that protein sample. But there are many areas, if you look at those areas, there is a no stain majority of the area, and that is one of the reason you cannot distinguish your protein molecules very well. Wherever there is a protein, little bit stain is deposited in the surrounding areas like this one, and that is the reason you can isolate that protein molecule. But overall TEM grid, it is not well stained the entire area. Only there is a isolated areas where there is a proper stain, and that's why we can somehow see the sample. So if we just consider this kind of images, definitely this is not a good image. So this will come. Okay, so we can see our protein sample and we can isolate the single protein molecule, there is no problem. But overall the image quality, if someone will ask, definitely this is a very bad quality images. So if we are getting this kind of images, it is better not to consider this kind of images seriously. It will be better to find out a little bit thicker stain area. So thicker stain area means there will be more stain will be deposited there, and then there will be a proper scattering difference between the protein and your background, and you can isolate your protein molecules properly. Okay, so now next, so this one is the thin sample, thin staining TEM images. Now we will just go to the thick stain region. So here you can see I am showing you again the same sample, but from a different area, where you can see the background is really. So if I just consider little bit lighter color, you can see these are the background, right? So this background is basically very thick background. Even if I just consider this is the another area, so even this area also. So overall this background is very thick, there is a too much stain is deposited, and that is the reason it is looking like this kind of black. And but in some cases you can see some triangular shape. So you can see some triangular shape, it is not very prominent, but still we can get some kind of protein molecules like that. You can see from this one it is very much identical. If you focus on this one, it is very much identical with this one. So this kind of thick stain, still you can isolate some particle, but what about there are so many other particles are there. So here you can see there are some particles are there, but we cannot see any proper shape of that molecule. So when we have this kind of staining, sometimes it is very difficult to isolate your actual protein. This entire image you can see there is a 4, 5 or 6 this kind of good particle you can see. Rest of the places it is just looking like a circular blob kind of structure. So if I just compare this staining solution again, what is happening here? So if we recall our negative staining technique, so what do you have? There is a carbon, okay, and then on top of that I have my sample here. My sample is here, but the problem here that we use too much strong staining solution, maybe. So generally 1% to 2% uranyl acetate and uranyl formate we should use it. So either the concentration is very high, or whenever we incubated our sample with the staining solution, we incubated the sample with staining solution for longer time, or when we blotted the sample, the blotting was not done properly, and that is the reason whatever staining solution was there all are deposited here like that. So this is my staining solution is deposited here. Now when electron beams are scattering from the background as well as the from sample, the scattering is very strong. So all are scattered in a very high angle, and that is the reason entire image is appearing, this entire image is appearing as a very black or dark image, and we cannot isolate our protein sample properly. This kind of staining, it is called a thick stain. of this negative stain. So, if there is a thick stain, this is a thick stain sample. If this is a thick stain sample, definitely it will be very difficult to isolate our protein sample and visualize our protein sample properly. So, this is, if I just consider again this one, if we just consider this sample, again this is a bad negative stain. Image. So, this kind of image we also should not consider for our any other purposes. So, let us take a look in the next image. So, here I am showing you another two different types of image. So, again, so this image is the same last image, that this is a thick stain image. So, in this thick stain image, if you recall that, okay, there are some good particle, you can see it, right. So, if I just go here, you can see that there are some triangular shape good molecule is there, but same time there are many particles are there. If we just consider, those have basically no proper shape, and this is happening because of this thick stain. We cannot identify any proper shape from those molecules. It is looking like a spherical or circular things. So, most of the time if there is a very thick stain, we will see this kind of pattern all over, and from there we can misinterpret our data that, okay, my protein is looking like a globular shape or looking like a sphere, but basically this is artifact is generating because of this high stain or very thick region we are trying to image. So, from there, here we cannot identify our particles properly, or we cannot see the morphology of our sample properly. So, this kind of data is again we will consider as a bad data or bad image. Okay. Now, we are coming to the next one. That next one is, there is a two possibilities. You can see there is a lot of large circular molecules are there, and we cannot identify any good molecules from here. Even if you pay attention, there are many places there is a circular rings are there all over. So, this kind of image, from here we cannot see any good particles. So, there is a two possibilities that we can see in background, there are so many things are there. So, it may be possible this protein sample is very concentrated, concentration is very high, as well as it absorbs lot of stain, that uranyl acetate and uranyl formate, and because of it absorbed lot of staining sample, this entire area appeared as a very thick stain and black. So, this kind of images are not good for our research purposes or any kind of work. So, if we have this kind of images, it is better not to consider those images for further studies. So, it will be better to not reconsider these things, go for the next sample preparation again. Now, I will just show you one more images, that, okay, there are couple of more images here I am showing you. In this particular image, what you can see, there is a white background. Most of the areas, if you look at these areas where you can see there is a lot of white backgrounds are there, huge amount of white backgrounds are there, but there are some places some black patches are there. So, this is basically staining is not proper at all. In this case also the same problem, that you have a white background, the huge, and then some protein samples or something is there which absorbs lot of stain. So, in this particular case, sample basically absorbs stain and your background staining is not correct. So, you can see this may be your sample, this may be your sample which absorb lot of stain. If you just come here, this absorb lot of stain, but your background didn't absorb any stain, and that is the reason we cannot isolate our sample from the background, because background is a light stain area. So this is basically our background. So this is basically our background and this is a light stain area. So we cannot isolate our sample properly from here, and that is one of the reason that this kind of stains are not good stain at all, and this is called the positive staining, because your protein sample absorbs some kind of black stain and the background didn't absorb any stain. So it may be property of that protein molecules is absorbing stain and it is not allowing the background to absorb the stain, or maybe this is a handling issue, or when we will prepared our grid, the grid is not good, and that is one of the reason the images are not looking good at all. So this kind of image we can always consider that this is not not an good image. So definitely this kind of images we should not consider for further study. So I'll stop it here. In the next class we'll discuss more about that light stain and thick stain, as well as I'll show you some of the good images, so you can distinguish that what is bad image and what is an good image. Thank you all for your attention.